there are three main things that come from the ectoderm. The first is your skin, essentially, the outer covering of most organisms, and everything that's part of that. That's not just your skin, but all the accessory organs, the glands, the hair, the nail, part of your eyes and things of that sort. There's all sorts of derivatives from that. So it's not just the skin, but all of the components that are in the outer layer. For chickens, it would be their feathers, all of the essential things that come to that outer layer. The second thing that comes about, and this is going to be in the next lecture, are neural crest cells, which, interestingly enough, even though they are derived from the ectoderm, they are almost considered a fourth germ layer, because neural crest cells don't only just become cells of neurons and skin or whatnot, they also become part of mesoderm and other things. In fact, they have a potential that is still retained that the three germ layers lose, which is why they typically call this the fourth germ layer, is these cells. But in terms of their history, they are derived from the ectoderm. So they are from that germ layer. That will be a lecture in and of itself. That'll be the next lecture on neural crest cells. Just an entire lecture just on that. And then the neural tube, which basically includes your brain, your spinal column, all motor neurons, a lot of the peripheral neurons, not all of the peripheral neurons, but a lot of the peripheral neurons that are going to be associated with that. Okay, so a lot of what we're going to cover today is about the neural tube, about neurulation, about how the brain forms, how the spinal column forms. We will talk somewhat about the skin and how that forms, but most of the focus is going to be on how the brain and the spinal column form. All right. Now, there are two forms of neurulation. After you start gastrulation, and as the three germ layers start forming, the ectoderm will then split off into two main areas. It'll be the ectoderm that's going to become the skin, and then you'll start the formation of what we call the neural plate. So the neural plate is a group of a part of the ectoderm that's going to be induced by the organizer that will take on a different fate than the rest of the ectoderm. It receives various paracrine signaling that allows it to invaginate and start forming a long tube, which we call the neural tube. So there's two forms of this process of forming the neural tube. The first is called primary neurulation, and the second, secondary neurulation. Primary neurulation is how most of the neural tube forms. Secondary neurulation is usually in the most posterior region of a developing embryo. So let me explain the difference between these two. Primary neurulation, the neural plate, or the part of the ectoderm that is differentiating to become the neural tube, is first going to start invaginating. Now, how it invaginates is actually there's a group of cells called the medial hinge plate that changes parts of its cytoskeleton and actually bends the plate in half and starts this invagination. It's not really a true invagination in that you have cells dipping down in. It's more of a folding up of this, but we do call it invagination. As you can see, it's forming a pit in that regard. Now, this is not the same pit as, say, the primitive groove that forms in the early chick gastrulation. This comes after Hens's node regresses back to the posterior end. Then you start getting the neurulation and the folding of the neural plate. So here you can see a cross-section of the upfolding, of the invagination. Right here is where we have the medial hinge plate. At a certain point, you get the dorsolateral hinge plates that then fold even further and further push these folds to where they'll connect to one another. You can see that right here. The dorsolateral hinge plate, you can see that right here, actually redirects the invagination and causes these to come together to eventually close off. So first you get the cells right at the medial hinge point that are going to change the cytoskeleton and cause the invagination, and then you get cells at the dorsolateral hinge plate or a hinge point to push them in even further. Here's where things start getting interesting. This convergence, where these two ends meet up, ultimately cause all of the neural plate to invaginate and then eventually pinch off and form a neural tube. And then the remaining ectoderm, that's what's going to form the skin and the other tissues that are overlying. So that's not going to become neural tissue, this epidermis region right here. This is going to become the skin, and for us, it's going to become hair and the sebaceous glands and all of those, whereas this is going to become the brain and the spinal column. Now, this is what we call primary neurulation, the infolding, the invagination, the convergence, the closing off, and the formation of the neural tube. So how is secondary neurulation different? Well, secondary neurulation, you have all of these free mesenchyme cells that are still neural tissue, but they'll coalesce and form epithelial tissue, and then they'll kind of hollow out the middle to form a neural tube. That's totally different than primary neurulation. But this only happens in a very, very small portion of the embryo, just the most posterior region of the formation of the neural tube. So here you have two completely different processes. One takes a plate, folds it inwards, closes it off, and creates this tube. Another one takes these free cells and coalesces them together and then hollows out the middle and forms a tube. Eventually, these two connect together, and they form one long continuous tube, and you can't tell the difference between the two. Some interesting things that we're still trying to discover about why secondary neurulation occurs in this most posterior region in this fashion rather than the entire neural tube forming just through primary neurulation. So here's looking at the chicken, since it's one of the easiest ones to look at neurulation. Here's the Henson's node. Here's as Henson's node is regressing, then you have the neural plate starting to invaginate. So here you can start seeing, here's the Henson's node right here. You can actually start seeing the invagination of the neural plate here. As we move further on, here are the neural folds folding up. So it hasn't quite closed yet. You get Henson's node still moving backwards here. And then eventually, this is showing in a much later stage the closure of this. We're going to talk about these various folds, because this is what the neural tube is eventually going to become. This is the brain. This is what's going to form the brain. And here's the spinal column right here. Okay, it's still open at this point as it's still closing off. It almost zips itself back up. In chickens and in other organisms, it kind of zips itself up. In humans, it actually connects at a couple points and then just kind of closes together. So it's not exactly the same in chickens as it is in humans. Let me see if I can, yeah. See, in humans, there's just a couple of hinge points that will close off, and then it'll eventually zip all together and completely close off the spinal column. You can see a couple points right there, and there's some gaps right in there, and eventually those will seal off. So it's a little different than in chickens. Chickens, it actually just zips up. It starts here, and it just kind of keeps closing up all the way back until it reaches the most posterior end. Here's where secondary neurulation occurs, right in this region right here. So primary neurulation is going to occur all the way back here till right about there, and then you're going to get secondary neurulation where the mesenchyme is going to coalesce and form the neural tube, and then connect up to the primary neurulation of the neural tube. You can actually see it kind of right here in that part too. So here you're seeing the neural folds. They haven't closed up yet. Here's a little more posterior. Here's even more posterior. So in fact, it kind of just forms as a wave. If you look more anterior, you're going to get much further primary neurulation. As you look more posterior, it's going to be farther behind, so to speak, in its folding and its primary neurulation. That's because of Henson's node. It goes from the anterior to posterior. So as it's going backwards, you're getting primary neurulation occurring more anteriorly, and then eventually it's going to catch up as Henson's node regresses backwards to the posterior end. So it doesn't happen all at once. That's why it kind of zips up from the anterior to the posterior region. So here's the most posterior region. Here's where you get some of these cells that are eventually going to coalesce into mesenchyme, and here you get these neural folds that will undergo primary neurulation. So it's really just the most posterior end that's going to undergo secondary neurulation. So from a side view, again, same process as what's happening in the chicken. You're getting the invagination, you're getting the infolding, and eventually closure off in the neural tube. So same process occurs in each one of these organisms, just slightly different as far as, whether it's surrounding an entire yolk or whether it's, the zebrafish that is undergoing its initial movements. But the same process of primary neurulation occurs in xenopus and in fish and in humans and in chickens and whatnot. So here's a human. This is showing, again, the same process of primary neurulation. What's different about humans is that it doesn't zip up from front to back. It closes up in a couple of different places, and then they eventually all seal up. And that might be one of the reasons why we're fairly prone, amongst the things that we talked about, to spina bifida is because it doesn't necessarily follow that zipping up. It kind of pinches off in a couple places and then eventually seals up. What I want to illustrate, too, is that in all these cases, there's always kind of an opening to the neural tube, an anterior neural pore and a posterior neural pore, which means that after the neural tube closes up, it's still open on this end and it's still open on the other end. It's open in both ends all the way to the end, and I'll explain why that plays a role here in just a second. So, under normal conditions, when the neural tube closes, then things will develop fairly normal unless there's other problems. Anencephaly is when there's a failure of the closure of the neural tube in the head region, whereas spina bifida is a failure of the closure of primary neurulation in the spinal region. And as I mentioned, there's various factors that go into there. Here's an in situ hybridization showing you the expression of the mRNA of the folate-binding protein. It doesn't actually show the receptor, but it's the protein that binds to the folate receptor that's necessary for closure of the neural tube. You can see that it is right in the region where the closure of the neural tube will occur. So here's the neural tube. This is the ectoderm overlying it, and there's where the folate-binding protein is found. Okay.